So good morning students. In the last class we were discussing about the codominance. Okay. So here the blood group in that codominance where both the alleles are going to express themselves, that we are calling it as the codominance. Okay. What is codominance? That both the alleles in a particular gene, both the alleles are going to express themselves, that we can call it as the codominance. So for that codominance, what example we had taken here for that codominant group in human being. This example we have taken where the genes IA and IA, these are the two alleles which will give a particular blood group, that is the A phenotype. This will be the genotype, this will be the phenotype. Then A, IA, IB. I means the gene responsible for the blood group. So here the blood group will become the AB, because A, B, these both genes are going to express themselves. Here, if A is dominant, then B is not recessive. B is also dominant. Both are going to express it. One more condition, IA and small I. This is the recessive gene, which does not have any sugar polymer. Okay. So here the blood group will become A. In this sense, we had studied about the phenotype and genotype of what? ABO blood group. So if see ABO blood group, here it is also, we can take this also as an example for multiple alleles. So what we can take? We can take as the example for multiple alleles. So let's see what do we mean by this multiple alleles. Okay, here let's see. Multiple alleles. It is a condition in which two or more genes. It is a condition in which two or more genes influence a single trait. Condition in which two or more genes influence a trait. Trait means that it is the character. Okay, two or more genes influence a one character, single trait in an individual. Here it is single trait of character. Single trait of character in an additive manner. Okay, here one more thing we can also write. There are more than two, that is three alleles governing the same character. One more meaning we can write here. There are more than two. There are more than two. More than two means that is three. Three alleles governing the same character. Three alleles governing the definitions, two definitions of what? Multiple alleles. So, since an individual, if two alleles are there, if two alleles are present, which are governing, which are meant for the single character, then that alleles we can call it as the multiple alleles. So here it is a condition in which two or more genes, that is either more than two genes, that is three genes. Three genes influence a single trait. Okay, single in an additive manner. They are going to add here for this ABO blood group. So in this ABO blood group, IA, IB. Okay, IA and IB. These are the two alleles. Okay, which will give a same character. What is that character? What is that phenotype? One blood group it is going to show, and that single character is the AB blood group. One more example you can take. IA. This is one allele, and this is the second allele. They both are meant for a single character. What is the character? That is the A blood group. Both are present in a form of additive manner, and that alleles are associated with the single character. That we can call it as the what? Multiple alleles. So sometimes they will ask for 2 marks. What do you mean by multiple alleles? More than 2, that means 3 or 4 like that. That much of allele is meant for only one character. That we can call it as the multiple alleles. So here one more example is there. A flower might appear in either orange or red or yellow. See here. On the basis of Mendel's work only, one more example is there. What is that example means? A flower is there. Example, this is one flower. And this flower, it may appear either orange or yellow with no graduation between the color. Either orange or yellow. There is no mixing up of orange or yellow. Separately, either it will be orange or either it may be yellow. By observing and studying plants and the methods by which they mutate, early researchers discovered more about the gene which determined the flower color. However, it was known that single gene, here you see, for the color, this is the, what is the trait here of the particular flower? That is the color of flower. This is the trait of character. Okay, so here capital O is meant for orange color. Then small o is meant for yellow color. Okay, if they get mated, if this is the male flower and that is the female flower, means that you see capital O, small o. They both are dominant. Okay, they both are dominant. So some of the flower of next generation, color of the flower will be orange or yellow. That means more than 2 alleles are responsible for a single character. The next, this is about the multiple alleles. Next topic is inheritance of 2 genes. That will be seen here. Before going to that inheritance inheritance of two genes. One more example they have mentioned in NCERT book that we have to see here. See here, a single gene product may produce more than one effector. So see here, this is also about multiple alleles. What they are trying to say here is a single gene product. Single gene product. It may produce more than one effector. One effector. One effector. So here, what example we have taken here means what example we have taken here means starch synthesis in pea seeds. Okay, see, pea seeds are these seeds are vegetables. Okay, they are going to store a carbohydrate or sugar molecules. What is that sugar molecule here, or what is that carbohydrate? That is the starch. So example here, starch synthesis in pea seeds. What is the example we have taken here? That is the starch synthesis in pea seeds. In a pea plant, see here, this is the pea plant, and this is the pea. In this pea, there will be presence of what? Seeds. Okay, so here these seeds are going to synthesize the starch. It synthesizes the starch. Okay, see here in these seeds, some seeds will be the larger and some seeds will be the smaller. Yes, sir. All seeds are not same there. Some are smaller, some are larger, some are medium size, like that it will be. And one more thing, some seeds will produce more starch. Okay, some seeds, what they will do? They will produce more starch. Some seeds will produce less starch. Okay, some will produce the more starch, some will produce the less starch. Okay, some will be the larger one, some will be the smaller one. So, which gene is responsible for the production of less of starch? Okay, so that we have to see here by taking here as a gene product which is producing a particular product. What is that product? That is the starch. From where? From the pea seed. So, this starch synthesis in pea seeds, it is controlled by one gene. Okay, this is one of the character. What is this? This is one of the character or trait. In what? In the pea plant. So here, it is controlled by one gene. Each character is controlled by the specific gene. So it is controlled by one gene. How many genes? For this one character, one gene will be there. Okay. The next thing, this one gene will have the two alleles. Okay. One gene will have two alleles. Here, this one gene, it will have the two alleles. And what are the alleles? Here we are going to represent it with the capital B. Capital B and small and small. These are the two alleles is present in one gene. That gene is responsible for what? For controlling that character. So, starch synthesis effectively means the more starch when it is which gene is going to produce. If it is BB homophile obviously they both are dominant. Capital B and capital B, they both are dominant and they are present in homozygotic condition. Homozygotic condition means here you see these are the parents. Capital B, capital B. Okay, if they mate, what will be the offspring? Capital B and capital B. They both are dominant. Yes, sir. They both are expressing themselves. Okay, so here that condition we can call it as the homozygote condition. So here the starch synthesis is much effective. In which condition? In this condition, starch synthesis is more effective. Starch synthesizes effectively. Okay, starch synthesizes effectively by what? By the capital B homozygote, and they will produce the large starch grain. Okay, produces large starch grains. Okay, then this is one more condition is there. If the alleles are capital B and capital B in a gene for the production of the synthesis of starch, okay, it will be having, and they both are also in which condition? Homozygotic condition. Homozygote condition. Here the starch synthesis is more effective, or the starch is going to produce more. But here, where they both are recessive, as we are representing it with small letter, that means it will be the recessive one, and they produces. See here, there is a lesser efficiency in starch synthesis. Lesser efficiency in starch synthesis. If there is a less amount of starch is going to synthesize here, what is going to happen means they produces the what they produces? They produces the smaller starch grains. Okay, which see here, one condition is there, capital B in a gene, two alleles are there, the two alleles are same. Here also one gene, two alleles are there, that two alleles are also in homozygote condition. Okay, so if these both are dominant, both are going to express more amount of starch, or starch synthesis is more effective. Both are recessive means lesser efficiency or less effective in starch synthesis. If more starch is going to produce, the size of the seeds will be larger one. If it is going to produce less amount of starch means the the seeds size will be the smaller. So these are the two conditions. After maturation, what happens? After maturation, what happens means whatever the seeds with the capital B genes, okay, the seeds with the capital B genes, that will be the round one, and the seeds with the small b, it will be the wrinkled one. Okay, round and wrinkled one. So here, this is about only the homozygote condition. Homozygote. One more example we can take to study the what happens if this homozygote is in place of homozygote condition, if it is a heterozygote condition. Okay, heterozygote condition means capital B small, like this, in this way. Which type of cells are going to produce? How much starch efficiency will be there? That we will see here. In a heterozygote condition, okay, it produces the round figure. Heterozygote condition, what will be the alleles? Capital the round figure. Heterozygote condition, what will be the alleles? Capital B, small. Where capital B is the dominant one. Okay, where capital B is dominant and small b is recessive. Okay, so here the capital B, which is dominant, they produces the round seeds. Capital B, which is dominant, it produces the round seeds. If it is round seeds, then what will be the efficiency of starch? It will be more efficient in starch synthesis or starch grains. Starch grains means that is the pea seeds. Okay, so what happens? Starch grain produce here it will be of intermediate size. Intermediate size of starch grains are produced. But it is more efficient in starch synthesis. Why? Because it has a one dominant gene, that it is the capital B, due to the incomplete dominance. B when it large size or round seeds. Okay, in that conditions, what they will produce? They will produce the medium size. This will become the intermediate or medium sized starch grain. Not too much large, not too much starch, because they both are going to express. Okay, so this is about what? About the starch grain size. We can consider it as the phenotype. So here the alleles, these alleles will show. Allele shows which dominance? That is the incomplete dominance. Okay, so this is about what? About the explanation about the multiple alleles as well as the incomplete dominance. We are taking an example of what? Starch synthesis in pea seeds. The next we will go for the inheritance of two genes. Next, inheritance of two genes or dihybrid cross, which is very important question for 5 marks. Okay, inheritance of two genes. Two genes are dihybrid cross. Which is very important question for 5 marks. Okay, so here a cross between two homozygous individuals. What is the definition of this dihybrid cross or inheritance of two genes? Means a cross between two homozygous individuals differing in two pairs of contrasting characters. Differing in two pairs of contrasting characters. This is the definition of dihybrid cross. Here, a cross is going to be made between two individuals. Okay, between two individuals which differing in two pairs of contrasting characters. Okay, the two pairs of contrasting character here, that is going to inherit to the next generation. So that we can call it as the inheritance of two genes or dihybrid cross. In monohybrid cross, only one gene is going to be inherited to the next generation. Only one gene, which is responsible for one character. Where in monohybrid cross. But in dihybrid cross, in di means two. Okay, here a cross is going to be made between the two individuals. Okay, differing in two pairs of contrasting characters. Two characters I am going to take here. Two characters. That means two genes. Okay, so that we have to see how they are going to cross. How that two genes are going to inherit to the next generation. See here. First we will take the parents. Okay. Parents. What characters they are going to take here in the parents? Two characters they have to take, differing in two pairs of contrasting character. Okay. That is seed shape and seed color. What are the two characters here? Two pairs of two contrasting characters means one is seed shape and one is seed color. Seed shape here, what it is means, it is round and contrasting opposite to this, that is ring color. Otherwise, I will write here. What are that? Two characters or two contrasting characters means, that is, one is seed shape and another is seed color. Two contrasting means opposing characters. Okay, one is round and wrinkled. Round means the seed will be like this. Wrinkled means the seed will be like this. Okay, seed color means yellow and green. Okay, these are the two characters which are going to inherit from one generation to another generation, or parents to F1 generation, then from F1 generation to F2 generation. Okay, so let's see here. What are the two contrasting characters? Seed shape and seed color. Round, wrinkled and yellow, green. So let's see here. Parent 1, it is round yellow. And parent 2, it is wrinkled green. Round yellow. In parent 1, these two are the characters. Okay. In parent 2, these two are the characters. In parent 1 have the character or the seed is round and yellow in color. And seed 2, parent 2 have the seed which is wrinkled and green in color. Okay. So see here, round yellow means we can represent what are the genes or alleles means. Capital R for yellow, alleles means capital R for yellow, capital Y. Okay, two alleles for round, two alleles for yellow. Crossed with the wrinkled means just they are contrasting. Opposite to capital R, it will become smaller. Opposite to yellow, that it is green, we can write it as the small y. Small y. Smaller, small y, small. Okay. Now next here, this is about the genes of alleles. During gamete formation, they are going to separate. Here you see gametes. During this time, what happens means this will get separated. Capital R, capital Y. From one pair, only one gene we are going to take. Okay, crossed with the small r and small y, like this. Okay, due to the law of segregation, during gamete formation, the genes are going to separate. The next, if we cross them like this, we will get the F1 generation. See here, F1 generation. Here in F1 generation, what will be the genes? Capital R, small r, Okay. Then capital Y, small. This will be the offspring. RR, we have to take it one side. YY, we have to take it one side. During when? During the F1 generation or during the production of offspring. So what it will become is, so here it will become, which one is the dominant here in between the capital R and small r? That it is the round. Capital R is the dominant one. Okay. And small r is the recessive one. So we can write it as the round. In between these two, capital Y and small y, yellow and green, which one is the dominant means capital Y, then we can write it as the yellow. So whatever the offspring after with the round yellow with wrinkled green, what we get the offspring, that here it is round yellow. Okay, two genes are going to inherit to the next generation, that we can call it as the inheritance of two genes, and that two genes will have the two pairs of contrasting characters. Okay, the next step after the F1 generation, that is the self-cross. How to do self-cross? So let me see. Self-cross, in that we have to take these genes of offspring, capital R, small r, capital Y, small y, cross with the capital R, small r, capital Y, small y. Okay. Otherwise, capital R, small r, into capital Y, small y. One more time. Self-cross means we have to take the same genes with the same plant. Okay, means the plant which has the same genes or same characters. With that, we have to self-cross. Capital R, capital Y, into small r, small r. Okay, so here we have to cross in this sense. Cross means we have made here cross like this. Okay, so it will become capital R, small y. We are going to cross these two genes with these two genes. Okay, where the capital R we are crossing with the capital Y. What we will get here? Capital R, capital Y. And again, capital R, small y. This one capital R with the small y. Okay, then small r, capital Y, then small r, small y. In this sense, we have to cross this one as well as that one. Okay, that it's not possible to visualize here, so I will show you here. Capital R, small r, into capital R Y, small r. Once again, okay. Become capital R, small, capital Y, capital R, small y, small r, capital Y, then small r, small y. In this sense, we have to cross. So we got now the male genes and the female genes. Now we can go for the production of F2 generation. So that after completion of self-cross, that is about the self-cross, the offspring what we got in F1 generation, we had crossed with the same offspring. Okay, then we have to go for the F2 generation. So let's see about the F2 generation. Here you see. So we have to take that gametes over here. What are that? Capital R, capital Y, capital R, small r, capital Y, and small r, small. These are the gametes of one parent. Then we have to cross with another parent and their gametes. That it is capital R, capital Y, capital R, small r, capital Y, then small r, small y. Okay. Now next we have to cross here. Cross this one with this one. Capital R, capital R, capital Y, capital Y. Then this with this gamete. In that capital R, capital Y, small y. Here it is small y. Okay. The next capital R, small r, capital Y, capital R. Now next we have to cross this one with that one. Fourth column. Capital R, capital Y, small y. Then next come to the next row. Capital R, capital Y, small y. Next capital R, small y, capital R, both are dominant. Here also both are dominant. So it will become round yellow. Okay. Here these both are dominant, so it will become round. But here you see one is dominant, one is recessive. We have to mention which is the dominant, not recessive one. So it will become round yellow. Okay. The next here also in both of these genes, small r is the recessive one, but we have to mention about the dominant gene. So it will become round yellow. Same thing, round yellow. The next here you see, this also will become the round yellow, because here they have given capital Y, small y. Capital Y is the dominant one. That's why you have to mention as the yellow. Capital R, small y, they both are dominant. It is round. But here they both are small y. They both are recessive. Then we have to mention the which color it will be. It will become the green. Round green. Okay. The next round yellow. Then round green. Here it is. Both are recessive. So it will become wrinkled. Wrinkled yellow. Here it is. Both are small r. Wrinkled yellow. Here it is round yellow. Next, here is it's also round yellow. Round yellow. Then again it is round green because they both are small y. It's round green. The next wrinkled yellow, round green. The next wrinkled yellow. Both are wrinkled green. Now next we have to find out the phenotype and genotype ratio. Here you see phenotype ratio and genotype ratio. In this one you see phenotype ratio means we have to see only the characters. Round yellow 1, round yellow 1, round yellow 2, round yellow 3, round yellow 4, round yellow 5, round yellow 6, round yellow 7, round yellow 8, then round yellow 9. Okay, so 9 round yellow color seeds are produced during the F2 generation. This is the F2 generation. Okay, so we can write it as the 9. Then next one. After round yellow, one more character is there. After round yellow, one more character is there. Round green. Round green 1. Round green 2. Then round green 3. Okay. Then it will become 9:3. Okay. The next one. Wrinkled yellow. Wrinkled yellow 1. Wrinkled yellow 2. Wrinkled yellow 3. 9:3:3. Then remaining one, this is the wrinkled green. Only one is there. 1. Okay. So this is about what? About the phenotype ratio. Next comes the genotype ratio. Okay. So here in this genotype, we have to see only the genes. Okay. So see here. Like these, how many are there? We have to see. Okay. So, like this genes here, it is capital R, capital Y, capital Y. Only one is there. This is 1. 1. Capital R, capital Y, small y. How many are there? That we have to see. 1. Then this is the 2. 1, 2. Okay. 1:2. Then next here capital R, small r, capital Y, capital Y. How many are there like? 1. Then 2. Then 2. Then comes capital R, small r, capital Y, small y. 1. Okay. Then 2. Then 3. Then 4. Okay. 1:2, 2:4. Then again, here you this one. Capital R, small y, small y. 1. Then only one is there. 1. Then capital R, small y, small y. How many are there? 1, 2, 2. Then next comes here. Smaller, capital Y. 1, only 1. Okay. Smaller, capital Y, small y. 1, 2. 2, then last one, 1. 1 is 2, 2 is 2, 2 is 2, 4 is 2, 1 is 2, 2 is 2, 1 is 2, 2 is to 1. This is the genotype ratio. 9 is to 3 is to 3 is to 1 is the phenotype ratio. And this checkerboard is very important. If you have any doubt, you can ask in the WhatsApp chat. Okay. Thank you. Next we will continue the next class.